This section will cover the urinary system. Our urinary system helps to regulate our blood ions as well as the blood volume, blood pressure, and blood pH through filtering. It is also responsible for the productions of some hormones. This illustration shows the organs of the urinary system and their relative locations in the body. Compared to other systems we've studied, the urinary system is fairly small. The kidney itself is about the size of a fist and is located behind the peritoneal cavity, or retroperitoneal. It is the shape of a kidney bean. The outside is covered with a fibrous, tough renal capsule. The outer portion of the organ is the renal cortex, while the middle or inner portion of the organ is the renal medulla. Within the kidneys there are pyramid-shaped structures called renal pyramids. These contain the nephrons or filtering structures. The renal columns are the spaces between the renal pyramids. The renal pelvis is the center area where the nephrons will drain the urine into after it has been filtered. The jagged edges of the renal pelvis help the already sharp crystals that form in kidney stones to have pointy edges, making them even more uncomfortable to pass. The minor calyces are funnel-like structures which drain the pyramids in collecting ducts. These merge together to form the major calyces which drain the minor calyces. This illustration shows most of the major structures within the kidney. This is an additional picture of a model of a kidney showing each of these structures. The renal artery will carry blood to the kidney to be filtered. The renal artery will branch into afferent arterioles, which will each carry blood to one of many glomeruli. The glomerulus is the initial filter of the blood going into the nephron. After the fluid has already passed through the glomerulus, it is known as glomerular filtrate. The efferent arterioles will carry blood away after it has passed through the glomerulus, and these arterioles will merge to form the renal vein. The peritubular capillaries surround the loop of Henle in the nephron to help reabsorb fluid back out of the nephrons. We would have difficulty remaining hydrated without being able to recover some of the fluid that initially passes through the glomerulus to form the glomerular filtrate. The blood from the peritubular capillaries eventually joins with the renal vein. This is an animation of the vascular system of the kidney by Wisconsin Online. This link can be linked to by clicking on it here on the Angel, watching it through Tegrity, or it can be found on the website under the links for this classroom as well to be able to watch it. It does not look like it is going to play on my computer today. This shows the structure of the glomerulus surrounded by the Bowman's capsule. Some of the fluid in the plasma passes through the capillary walls in the glomerulus to form the filtrate, which is captured by the Bowman's capsule. The nephron is the main functional unit of the kidney that does the filtering. The renal corpuscle includes the glomerulus and capsule that surrounds it, or the Bowman's capsule. The renal tubule carries the glomerular filtrate after it leaves the glomerulus. This tube will change names as it moves along. The glomerular filtrate is filtered plasma after it has gone through the initial filtration by the glomerulus. Here it removes the cells and other large molecules. Glucose is removed up to about 170 milligrams per deciliter. If the blood sugar exceeds that level, it will spill glucose into the urine. The way that diabetes used to be tested for was someone would get to taste the urine, since diabetes is now diagnosed at 150 milliliters per deciliter. Our current tests are going to be more accurate at testing for diabetes since it does pick up at a lower level, but the urine would nonetheless have sugar spill into it at the higher levels and still be sweet if someone were to want to taste it. The Bowman's capsule or glomerular capsule is the structure that surrounds the glomerulus to capture the filtrate. This is an illustration of how a nephron sits within the renal pyramid. Each pyramid will contain numerous nephrons, and you can see how the shape of the nephron here can lead to the formation of a pyramid structure when you have several next to each other that are wider at the top and narrower at the bottom. The proximal convoluted tubule is the region of the tubule closest to the glomerulus that begins to take a curvy route. It will then extend down deep into the pyramid, forming the loop of Henle. The loop of Henle is surrounded by the peritubular capillaries to absorb fluid back into the bloodstream. After the loop of Henle, there is another curvy portion of the tube called the distal convoluted tubule. The distal end of the tube will join with the collecting duct. Multiple nephrons can drain into one collecting duct. The fluid is now urine, and when it enters the collecting duct, since the filtration process is completed in the nephron. The collecting duct will carry the fluid down to the calyces, where it will join the renal pelvis. The initial filtering process occurs at the glomerulus when making the glomerular filtrate. As the fluid passes through the nephron, we have tubular reabsorption that occurs to help pull water and ions back into the kidney. The kidney will pump the sodium back in, and the water will follow the sodium. This occurs in the loop of Henle. In the tubular secretion, some of the water and ions are put back into the urine as necessary. This filtering process occurs as the fluid passes back and forth across a membrane. This secretion takes place in the ascending part of the loop of Henle and the collecting duct. This is all driven by the concentration gradient formed as you move through the kidney. This illustration shows where secretion and absorption occurs in the nephron, as well as where various hormones will target the nephron. This illustration shows the concentration gradient as you move deeper into the renal pyramid, driving the fluid reabsorption and the secretion of filtration. You can see here as you move down into the area that's darker purple, what that represents is a greater sodium concentration. The higher the sodium concentration, the more it is going to pull water out of the loop of Henle, and it will go back into these peritubular capillaries. As you move back up, that concentration of sodium goes down, allowing some of the water to go back in to the loop of Henle. The RAA system, or renin-angiotensin-aldosterone system, will target the kidney. As you increase the sodium and water retention, urine volume will decrease. ADH, or antidiuretic hormone, will also increase water retention and decrease urine volume. Once the fluid is completely passed through the glomerulus, it is now called urine. Urine should be translucent and a pale straw color. In general, if the urine is darker, it will indicate dehydration. Some substances will color the urine and do not indicate dehydration, such as B vitamins and certain medications. It is also not unusual to see the urine be the color of maple syrup in elderly persons who do not consume enough water. While it's common that we will see it, it does not mean that it's a good, healthy situation. The ureters are the tubes that drain the kidneys into the urinary bladder. The urinary bladder stores the urine until a convenient time to release it. When lying supine, the urinary bladder will actually pinch off the tubes to the ureters to prevent the fluid from flowing backwards into the kidney. This also keeps any of the fluid produced in the kidney from getting into the bladder when this occurs. This is why when you get up in the morning, you will need to empty your bladder not long after sitting or standing up. The urethra is a tube which connects from the urinary bladder to the external body. Here we have another animation of the urinary system.
The major excretory organ of mammals is the kidney. Humans have two kidneys located in the upper rear region of the abdominal cavity. The urine they produce is conducted to the urinary bladder through the ureters. The urethra drains the bladder. The internal structure of the kidney includes an outer cortex and an inner medulla. The ureter divides into branches, the ends of which envelop medullary tissues called renal pyramids. The actual work of the kidney is carried out by functional units called nephrons. Each human kidney contains about 1 million nephrons. Each nephron consists of vascular and tubule components. An afferent arteriole carries blood to a knot of capillaries called the glomerulus. Draining each glomerulus is an efferent arteriole that gives rise to the peritubular capillaries, most of which surround the cortical portions of the nephron tubules. Blood pressure forces water and small molecules to be filtered from the glomerulus and collected in Bowman's capsule. The initial segment of a renal tubule is called the proximal convoluted tubule. The glomerulus, Bowman's capsule, and proximal convoluted tubule of each nephron are located in the cortex. From the proximal convoluted tubule, the nephron tubule turns down into the medulla. The portion of the tubule in the medulla is called the loop of Henle. Where the ascending limb of the loop of Henle reaches the cortex, it becomes the distal convoluted tubule. The distal convoluted tubules of many nephrons join a common collecting duct in the cortex. The collecting ducts then run in parallel with the loops of Henle down through the medulla and empty into the ureter at the tips of the renal pyramids. A few paratubular capillaries run into the medulla in parallel with the loop of Henle and the collecting duct and form the vasa recta. These capillaries carry away the molecules that are reabsorbed from the tubules. All of the paratubular capillaries join back together into a venule that eventually leads to the renal vein. Nephrons regulate the composition of blood and urine by a combination of filtration, secretion, and reabsorption. Viewed schematically, we will see how these processes are facilitated by the regular arrangement of segments of the nephron. The proximal convoluted tubule is responsible for most of the reabsorption of water and solutes from the glomerular filtrate. The cells of this section of the nephron actively transport sodium ions and other solutes, such as glucose and amino acids, out of the tubule fluid. The active transport of solutes out of the proximal convoluted tubule into the tissue fluid causes water to flow by diffusion. The water and solutes moved into the tissue fluid are taken up by the paratubular capillaries and returned to the venous blood, leaving the kidney. Despite the large volume of water and solutes reabsorbed out of the proximal convoluted tubule, the overall concentration or osmolarity of the fluid that enters the loop of Henle is similar to that of the blood plasma, although its composition is quite different. The ability of the kidney to produce urine that is hypertonic to the blood plasma is due to the loop of Henle. The loop of Henle does not concentrate the urine directly. Rather, it functions as a countercurrent multiplier, creating a concentration gradient in the surrounding medulla. To understand the countercurrent multiplier mechanism, it's easiest to move backward through the tubule, starting with the thick ascending limb. The thick ascending limb actively transports sodium ions from the tubule fluid and moves it into the surrounding tissue fluid. Chloride ions follow passively. The thick ascending limb is not permeable to water, so the reabsorption of sodium and chloride ions out of this part of the tubule is not followed by the outward diffusion of water. This reabsorption of sodium and chloride ions raises the concentration of solutes in the surrounding tissue fluid. The descending limb, in contrast, is permeable to water, but not very permeable to sodium and chloride ions. Since the surrounding fluid has been made more concentrated, water leaves the tubule by osmosis. As a result, the fluid in the descending limb becomes more concentrated as it flows toward the bottom of the medulla. The thin ascending limb is not permeable to water. It is, however, permeable to sodium and chloride ions. Since the tubule fluid is more concentrated than the surrounding tissue, sodium and chloride ions diffuse out. The thick ascending limb continues to move sodium and chloride ions to the medulla by active transport. As a result of this process, the tubule fluid reaching the distal convoluted tubule is less concentrated than the blood plasma, and the solutes that have been left behind in the renal medulla have created a concentration gradient in the surrounding tissue fluid. Since the fluid entering the distal convoluted tubule is less concentrated than the surrounding cortex, the tubule loses water osmotically as it flows toward the collecting duct. The tubule fluid entering the collecting duct is at the same concentration as the blood plasma. However, since sodium and chloride ions have been moved out of the tubule fluid, urea and other waste products make up a greater proportion of its total solute content. As the collecting duct descends from the cortex to the tip of the renal pyramid, the concentration gradient established by the loop of Henle increases. This increasing solute concentration causes more and more water to be absorbed from the fluid, thus concentrating the urine in the collecting duct.
The function of the mammalian kidney may be summarized as followed. The glomeruli filter large volumes of blood plasma. Most of this volume, along with valuable molecules such as glucose and amino acids, is reabsorbed from the proximal convoluted tubule. The loops of Henle create a concentration gradient in the medulla of the kidney by a mechanism called the countercurrent multiplier. As the filtrate flows up through the thick segment of the ascending limb, sodium chloride is transported out of the filtrate into the extracellular spaces of the medulla. The resulting increase in the osmotic concentration of the extracellular fluid draws water out of the filtrate in the thin descending limb, making it more concentrated. As this more concentrated filtrate flows up through the thick ascending limb, more sodium chloride is transported out, and the extracellular fluid becomes even more concentrated. Thus, the opposing or countercurrent directions of the flow of the two limbs of the loop of Henle, along with the differential permeability and transport capacities of these limbs, results in multiplying the concentration difference between the filtrate and the extracellular fluid of the medulla. The concentration difference is not uniform throughout the medulla, but forms a gradient that increases from the region next to the cortex to the tip of the renal pyramid. In response to this gradient, as urine flows down through the collecting duct from the cortex to the tip of the renal pyramid, the urine can lose water osmotically and become more concentrated. This countercurrent multiplier mechanism makes it possible for mammals to produce a urine that is hypertonic to their blood plasma. Mammals inhabit an enormous range of environments on Earth, including some of the most arid. The major adaptation that allows mammals to maintain homeostasis in the face of a wide range of osmotic stress is the variable ability of their kidneys to concentrate urine. Thus, when they take in lots of water in their food, they can excrete the excess water by producing dilute urine, and when they are exposed to very arid conditions, they conserve water by producing highly concentrated urine. The trigone is the inferior one-third of the bladder. This region also helps to detect and send the signal when the bladder is getting full. The urge will begin when the bladder contains around 150 milliliters of fluid. The external urethral orifice is the opening from the urethra to the external body and is under voluntary control. The internal urethral orifice is the opening from the urinary bladder into the urethra. This portion is under involuntary control. The internal urethral orifice is held closed at the internal urethral sphincter. This is a ring-shaped muscle controlling the sides of the orifice and is under involuntary control. The external urethral sphincter is the ring-shaped muscle controlling the external urethral orifice. This is under voluntary control, although the voluntary control is limited after the internal urethral orifice has decided to relax. These muscles often atrophy after menopause, contributing to the varying degrees of incontinence and difficulty holding urine at this point. Micturition is the process of urination.